Well, hello and welcome to the first RIEEE webinar of 2023. We are really excited to have all of you here and those who are able to watch back later for an exciting kind of lineup that's going to be showcasing some work that's already happening here, but also sort of laying an invitation for how we might grow that going forward. So I'm Christine Hendren, RIEEE Director, and we have some great presenters that will be introduced in more detail coming up. But we have Marta Toran and Matt Ogwu and Rachel Bleich, and all three of them are going to give a fantastic webinar today. I am going to just let you know the format is about 15 minutes per each speaker with a little bit of Q&A for them. But then if you're able to hang on for the half hour after, two of our speakers will be in a position to stay and could have a more in-depth discussion. So that is how we'll roll for the next hour or hour and a half. So, all right, great. Let me go ahead and share my screen. In this case, I'm going to start by introducing because I am, in this particular webinar, also a project member. And so I'll put a little bit of context on it as we jump in. And so our title today of our webinar is Convergent Science and Outreach. The ecology, biology, and mechanics of communication between bacteria, fungi, and plants in soils. So interkingdom communication by vesicle mediation is what we'll talk about. And we'll make that make sense for anybody who doesn't already do all of those words in your day job too. So the motivation behind it, so the reason it's convergent science is because this core research is powered by an NSF Convergence grant. And the purpose of that is to bring together and blend together different disciplines. So I won't go through this entire slide, but this is a nice, easy, as all analogies are, there's some flaws, but it's a nice, easy analogy for explaining what we mean by convergence. And really, sometimes you need disciplinary expertise if you're going to change a core theory, and we have that in this project. We need multidisciplinary kind of interactions as well, interdisciplinary on the spectrum. And then transdisciplinary goes more, more from integrating into actually blending them together. And so then that's when you get to the actual smoothie. You can't really tease out the pieces of the fruit anymore. You couldn't ask the questions you're asking without having blended together your methods, your ways of thinking about the problem. And we think of convergence as transdisciplinary work that brings in even maybe non-academic people into the fold so that you have everybody kind of on a shared vision toward use-inspired research. So we also say that, to make the smoothie, you need a blender. That's kind of the convergence team science angle that pulled this project together originally. And especially what that is giving is the lid. So you can't just like throw a bunch of disciplines together and say, how do cells communicate with each other in soils? You have to have some kind of constraints on what are the right things you should do together. And this team has done that over the past couple of years. So just very at a high level of what motivated this, we were coming as a team, and this is prior to the App State team that you'll meet today, coming from a background of nanomaterial fate transport and risk assessment in environmental systems. And what we realized was that predicting how very, very tiny particles in that size range would move from one place to another in the environment also ended up helping us predict what they would change into, what they would associate with, what would be the impact of these tiny particles. And then we connected with some people who were they were working on liposomes and in blood. And they were trying to do the same thing, figure out where these tiny, tiny particles in blood were going to go and based on what. And what we realized when we met was that the different disciplines and contexts that we were asking similar questions had different blind spots. So maybe on the bio side, they kind of understood the biology of the particles, the bio nanoscale particles they were dealing with much better than they understood the forces that were governing the transport. And so we thought, wow, what if we have answers to each other's questions already if we get in the right room? So this started as a Duke project. At the time, I was at Duke University, and you don't have to digest this entire Venn diagram, but we pulled together geosciences and biogeochemistry, biosciences and molecular cell biology with engineering transport, and then a lot of intersections in between using convergence and team science methods to try to ask these questions about how is it that, these cells generate messages for each other and somehow are able to get them to each other. And what is state to bring in an app state team. So myself, I came over and really grateful to have been here for the past 2.5 years. And we also got a lot more expertise in that middle ground. So the PI of the project here is Marta Toran. Then and that brings in the K-12 education and outreach type of expertise, which touches all the expertise. And then Matt Ogwu, who will present his work today, really sits kind of between things too, with ecology, biology, characterization, and variety of things he'll tell you about. And Rachel Bleich also kind of sits at another couple of intersections, working in microbiome work, touching environmental microbiology, and then also just cell biology. So it's really great to have added a big app state team, and we have the capability to expand, to get leveraged funds, and maybe ask some other interesting questions together and make a delicious smoothie. So that's all I wanted to do to tee this up. So I'm going to pass it back to Grace so that she can properly introduce our speakers and get to the fun stuff.
Thanks, Christine. So we have 3 speakers today. As Christine mentioned, they'll each take about 12 minutes or so, with maybe some time for a question as they switch between one speaker to the next. Our first is Rachel Bleich. She's an assistant professor in the biology department, and her research focuses on microbe, and host microbe interactions, particularly in the gut microbiome. And we have Matt Ogwu, who's in the Department of Sustainable Development, an assistant professor. Matt's research focuses on sustainable biodiversity and ecosystem management from the gut microbiota to landscape scales. And we have Marta Toran, who's a lecturer and outreach coordinator in the Department of Geological and Environmental Sciences. She teaches various environmental science and science communication courses and facilitates quite a few STEM outreach efforts, including the GeoBago Mobile Earth and Environmental Science Lab. So we'll go today in the order actually that I just introduced, starting with Rachel. So if Rachel, if you want to start sharing your screen, and then followed by Matt, and then Marta. And thank you all for being here today. Excellent. It's all yours, Marta, or Rachel.
Awesome.
Okay.
Great. Perfect. Thank you so much, Grace. I'm excited to get to share the first part of the project of what we've been doing here at App State. And so my piece kind of focuses on the isolation of the microbial extracellular vesicles for testing for interkingdom communication and soil systems here at App State. But to kind of, before I jump into a little bit more of that, so I'm a microbiologist and really think about how microbes interact with one another and their environment. And so I think historically, we kind of have thought about microbes as being more individual. We isolate a particular strain and kind of look at what it does and what genes it possesses. But I think in more recent years, we've really come to appreciate how important the interactions that microbes have with each other are in determining their behaviors, what types of molecules they produce, and just how they interact and what they do and what their function is. So right microbes can interact with other microbes or other members of their species or members of other microbial species. And then they can also, interact in these larger multi-species communities. We have multiple microbes, so bacteria, fungi, like we're also looking at with this project, all kind of in a larger group. And then, these microbes are also interacting with a host or with their environment. And so for this project, we were kind of thinking about this in terms of plant and soil systems, where they would have those other factors influencing how they're functioning. And in particular, we're thinking about these extracellular vesicles as signals that help mediate these interactions. So what do we mean when we're saying extracellular vesicles? These extracellular vesicles, or EVs, are these kind of cell membrane derived structures. So usually we think of them as kind of budding off from the membrane of the cell and getting released into the extracellular environment. And there's a large body of literature that shows how important these vesicles are for intercellular communication. And so pretty much all cells and all cell types will produce extracellular vesicles. But in particular here, I'm thinking about bacterial extracellular vesicles. So an example, on the left, we have a gram-negative bacterium. So it's got a double membrane, two membranes, an inner membrane and an outer membrane. Right? We can see these vesicle structures can either kind of bud off from the outer membrane or bud off that includes inner and outer membrane components. So, because they're budding off from the cell, typically, they'll hold, cargo from the periplasm or from the cytoplasm inside the cell. So the RNAs and other nucleic acids, a variety of proteins, lipids, and other small molecules. And then their membranes will also contain membrane-bound proteins that are on the surface of that cell. Right, and then those molecules become signals that get released out into the environment. And so in the kind of the system we're thinking about, so between bacteria, fungi, plants, there are definitely some positive and some maybe almost negative benefits for the bacteria and the plants from these vesicles. So a clear benefit, is these vesicles can actually help plant immunity. So plants will pick up vesicle signals from bacteria, upregulate their innate immune system, and help them defend against further pathogens. For the bacteria, these vesicles can be decoys for antibiotics. So if they release vesicles, the antibiotics will attack vesicles, leaving them maybe able to escape cell killing or cell death. It can be helpful by helping the cell export harmful molecules, so unfolded proteins or other things that it would like to get rid of. And it can help the cell acquire certain nutrients, Bacteria can use it to scavenge for iron or other things it may need that are more limited. On the flip side, for the bacteria, when the plants do upregulate immunity, that can also be negative if then they attack that bacteria and use their immune system to ward off maybe a bacterial invasion. And it also comes as a metabolic cost to the cell. So there is some energy they have to put in order to bud off these structures. And so what we're really kind of focused in, is how they're really functioning in communication. And there are a variety of ways this can happen. So if we're thinking kind of between microbe to microbe, there's a whole range of ways this can be signaling and communicating. So microbes are transferring DNA to share that genetic information. It's helping with quorum sensing, which determines bacterial coordinated behavior with things like biofilm formation. So what kind of phenotype they're going to be in. And also can be involved in predation, so as signals or enzymes to lyse open another bacterial cell to release those nutrients into the environment. And then right between kind of microbe to host or microbe to plant, again, it's this kind of stimulation of immune response, and even some like RNA interference, where those RNAs, small RNAs that get packaged into the extracellular vesicles can change the gene expression of the host cell. So kind of when we approached this project, we had, for our part, a couple of goals in mind. So what I kind of took on was isolating these extracellular vesicles from kind of some model bacterial and fungal organisms. And then, as you'll hear from Matthew Ogwu next, we then use those to examine the biological effects of these bacterial and fungal EVs kind of in the plant fungal bacterial interactions within the soil system, and can do further analysis on the plant-associated microbiome to see how that is influencing the community. And a lot of this work was done by a grad student who's been working with both myself and Matthew, named Will Harden, who is shown in this picture here. So for what we're doing at App State, we used 2 strains in particular. So a fungal strain, Saccharomyces cerevisiae, brewer's yeast that's pretty commonly used, but is also found in soils and in the environment. That was kind of our model fungus. And then we used a model bacterium, Pseudomonas fluorescens. This is a gram-negative rod-like bacteria, rod-shaped bacteria, that's also commonly found in soil systems, and is not itself a key pathogen for plants, but it's just something that's normally present. Awesome. And so the approach we took to do the isolation of these extracellular vesicles was a series of centrifugations and filtrations. And so we grew up our microbes in liquid culture. And we're just, for this experiment, we were just using a standard nutrient-rich culture. But something I do think about for the future is kind of how we manipulate those culture conditions, and change the nutrient profiles. How will that impact the EV production and ultimately impact the signals that they're sending? But we grew up our microbes, centrifuged them to remove the cells, so the bacterial cells or the fungal cells, and then did a filtration step to remove any lingering bacterial or fungal cells. So now we've collected kind of our supernatant that's a mix of EVs and other molecules and media components. And we do an additional centrifugation step at a faster speed to pellet out those smaller cargo, so those EVs, and then can collect those, further filter them, wash them a little bit, and concentrate them at a centrifuging with an ultracentrifuge. And then at the end, we do a quantification. So the main quantification we're using in order to know how much to apply in our systems is through Bradford protein assays. But we can also look at lipid concentration and some other metrics to quantify these EVs. And so for both the Saccharomyces cerevisiae, our fungus, and Pseudomonas fluorescens, our bacteria, based on that protein quantification, we had either 175 micrograms per liter or about 5 milligrams per liter of protein. And that was the metric we used to then dilute and apply to our plant soil system. And so, yeah, so for what I'm thinking about moving forward, so you'll hear from Matthew next about how that went with the plants, but my lab is really interested in how these interactions impact us as human hosts. So we sometimes think about different systems, so a lot of the same principles apply. And in particular, we're interested on the impact on inflammation in our gastrointestinal tract with inflammatory bowel diseases like Crohn's disease, which is chronic inflammation all throughout your GI tract. And one thing we've noticed with our, in some of the experiments we've been doing, is that a couple of bacteria tend to be very positively correlated with each other and with inflammation in the large intestine. And so it's an E. coli and Enterococcus faecalis. And so we've been doing similar type co-culture experiments where we've been growing these bacteria together on plates and in liquid culture and looking at how they influence and signal to each other. And so in this top part on the right, we have an example co-culture where we've mixed Enterococcus and E. coli together. And what we see is an increased kind of wrinkled phenotype or pattern when they're grown together, which we think is indicative of biofilm formation. And as we can see at the bottom, when we've run like a standard kind of microbiological biofilm assay, we see increased biofilm production in co-culture. So something we're thinking about is, now that we've been thinking about these extracellular vesicles as signaling molecules between bacteria, fungi, and plants, we're thinking about how they might be signaling in these microbe to microbe interactions, even within our gut system. So we're applying some of these same principles, isolating vesicles, and then growing E. coli and Enterococcus in the presence of the others' vesicles, just seeing how that changes growth and how that might change this biofilm phenotype, because we know that biofilm formation can be signaled through extracellular vesicle communication. So that's kind of some of the future directions we're heading in my lab, but I'll leave off there. So I wanted to just thank my lab in particular, grad student Will Hardin and undergrad Jake, who've been doing this protocol here at App State and isolating extracellular vesicles, collaborators that are here, and the Duke team, especially Maida and Hannah, who sent us our environmental strains and helped us establish the protocol here at App State for the isolation. And with that, I can take any questions if there's time.
Yeah, thank you, Rachel. And we do have time for a question or two while they switch screens. We'll have Matt queue his up. Do anyone have a question for Rachel? A quiet bunch on Friday. Okay, thank you. I think we'll roll right on to Matt.
Great, thanks.
Thank you so much. I'll be speaking on the physiological ecology of Pseudomonas fluorescens and Saccharomyces cerevisiae, extracellular vesicle-mediated interkingdom communication and regulation on Arabidopsis thaliana. Basically, I got the samples, I got the vesicles extracted by the Black Lab, and then they handed it over to me. The reason why we stuck with Pseudomonas fluorescens is basically because of some of its characteristics, which I'll highlight here. And it includes the fact that it's a gram-negative rod-shaped bacilli, and it is ubiquitous in the environment, especially in the soil. It is also a model organism for understanding communication within the soil system, as well as communication between different microbes within the soil system, as well as the plant and microbes within the soil system as well. Pseudomonas fluorescens is also non-pathogenic, and it has an antagonistic effect on several fungal as well as soil-borne diseases, which makes it a very good model for this study. And also it increases the availability of nutrients in the soil, which is one basic challenge in the world, which if we can address that, to reduce the use of agrochemicals for growing food. The reason why we chose Saccharomyces cerevisiae is that it promotes the production of plant growth hormones like indole acetic acid, as well as it increases plant root to shoot ratio. And it has also been used as a soil amendment. So basically, we're not introducing something that is new to the soil. Both Pseudomonas cerevisiae as well as Saccharomyces cerevisiae, Pseudomonas fluorescens and Saccharomyces cerevisiae, they can both be found within the soil system. And Saccharomyces cerevisiae has also been used as a biocontrol for economic plant diseases, especially the root knot nematode of cucumber. And it also promotes medaphic diversity as well as interaction within the soil system as well. and it has also been implicated in several mutualistic interactions with plants as well. With regards to the experimental design, I use 7 treatment groups containing 4 replicates each. And the treatment groups include the control, which is just basically soil without any EVs introduced into them. Then we have the high and low for both Saccharomyces and Cerevisiae and Pseudomonas, as well as a combination of Saccharomyces and Pseudomonas, both in high as well as in low concentration, so that we can actually see where the effect is coming from through our experiments. So we collected the soil. We decided to use the Songro soil, which is a common soil used in horticulture. The reason why we chose to go with this soil is that it doesn't have any additive within it, and then we also have the constituent of the soil basically freely available for everyone, so we can actually know where the impact we're seeing within our experiment is coming from. We subjected the soil to sterilization, basically to remove any remnant microorganisms within the soil. Then after that, we introduced our Arabidopsis thaliana seeds. Within it, the seeds were freshly harvested. Within 1 month of harvest, we used the seeds. The seeds were introduced into the sterilized soils. Then after that, they were stratified in the dark for a few days, then transferred into the growth chamber and the condition that is set up, that is described below the growth chamber over there, with EVs added every 2 weeks after the first week, basically to actually see if we allow the Arabidopsis thaliana to acclimatize to the growth chamber for 2 weeks. Then after that, we started introducing the EVs in the concentration that were actually shown earlier. Then after 16 weeks, the growth was commendable for both the Saccharomyces cerevisiae treatment, as well as the Pseudomonas treatment, as well as in combination of Pseudomonas cerevisiae, Pseudomonas fluorescens, as well as Saccharomyces cerevisiae. But with regards to the control, we didn't notice the same level of growth and development compared to when we introduce these vesicles within the soil. So that led us to ask what could be responsible and how. At the moment, we're trying to answer the first question, which has to do with the above ground mechanism for this growth and development that we've observed within the 6-week period that we grow our Arabidopsis thaliana in the growth chamber. And then we're looking at it from a morphophysiological standpoint. The other 2 questions, which has to do with the phylospheric as well as rhizospheric effect of this, as well as mechanism of this interaction or this communication between the soil, the plants, and the microorganisms. We're still working on answering them, and I would throw more light at the end of the presentation. With regards to the morphophysiological effect that we've looked at the moment, we looked at different physiological parameters in the plant to see what kind of effect the Arabidopsis, the introduction of these vesicles has on them. So we were able to connect this effect to the chlorophyll. There was significant effect on the amount of chlorophyll within this plant as a result of the introduction of the vesicles in the soil. But with regards to other morphophysiological parameters, we didn't see so much growth. So we looked deeper into this effect that we found, and we're able to connect it to the pyro ring of the chlorophyll. Basically, chlorophyll, as you can see from the structure, has 4 pyro rings. Then at the center, we have magnesium ion. The pyro rings has nitrogen associated with them. And could it be that because these vesicles have a high amount of protein, which has a high amount of nitrogen within them, that's why we are seeing that effect. And we saw the effect both in chlorophyll A and chlorophyll B, and the difference between them is not in the nitrogen, but rather in the arrow group that is attached to one of the pyro rings. So for the chlorophyll A, we realized that the chlorophyll effect was mainly coming from the low concentration of Pseudomonas fluorescens, as well as the combination of Pseudomonas fluorescens, as well as Saccharomyces in high concentration. The others were not significantly different from each other. And basically, low concentration was consistently high throughout the experiment, as we can see also for chlorophyll B as well. The same Pseudomonas fluorescens at low concentration of the extracellular vesicle also had significant effect that was basically different, interestingly also from the control and also from the low concentration of the combined extracellular vesicle at low concentration of Pseudomonas fluorescens as well as Saccharomyces cerevisiae. With regards to the total chlorophyll, we also found some effects. And notice again, the low concentration of Pseudomonas fluorescens actually had the same significant effects, which was revealed through the post-hoc comparison to be different from other concentrations that were used for the experiment as well. So this significant amount of chlorophyll that is induced by this extracellular vesicle, what is the benefits? Basically, the increase in chlorophyll levels has been connected to increase in biological functions as well as biological activities. So could it be that the introduction of this extracellular vesicle into the soil is actually causing some above ground increase in biological function and activities within the plant? And also increase in chlorophyll level has also been connected to the protection against several chronic diseases within plants. And also this increase in chlorophyll level above a certain point can also act as a form of anti-mutagen. Considering the kind of environmental changes that we are experiencing due to climate change, this might also be an important point to reduce mutagens within the environment as well. Chlorophyll has also been connected to plant productivity through photon absorption as well as electron release. But in the case of this study that we conducted together, we didn't see so much increase in photosynthetic activity. It would have been very interesting if alongside the increase in chlorophyll, we also found increase in photosynthetic rate as well. But we only found increase in chlorophyll, but not in photosynthetic rate. And also increase in chlorophyll level has also been connected to increase in the availability of nutrient for plant growth and development, as well as that has been connected to high nitrogen in the soil. So basically the extracellular vesicle that we introduced into the soil acted as a form of nitrogen amendment for the soil, because high nitrogen in the soil, when you add conventional fertilizer into the soil, it also increases the concentration of chlorophyll in plants. So basically our extracellular vesicle also act as a form of fertilizer, as amendments for the plant as well. So where do we go from here? We'll be looking at the rhizospheric as well as phylospheric effect of this extracellular vesicle, and we'll be looking at it through 16S rRNA as well as ITS sequencing. We've started doing some DNA extraction to this effect, and when we are done, we should be able to connect the effect that we found to as well taxonomic and functional changes within the soil, and see if this can be connected to the extracellular vesicles that were added into the soil. Most interestingly, since the work is ongoing, I would like to put out an open invitation there for anyone that is interested to join the project through certain stage or contribute to certain points. Is welcome to join it. I also want to add voice to acknowledging some persons that have made some significant contribution to this work, including undergraduates that are working with me, as well as graduate student Will Adin, Richard Bleich, as well as Duke Vesicle team. I'm very grateful for all the assistance I've received to be able to conduct this experiment in collaboration with you guys. So thank you very much.
Thank you, Matt. While Marta is queuing up her screen, we certainly have time for a question or two for Matt or Rachel, or the connections between the two.
I have a question, if that's okay.
Yeah, thanks, Chiquita.
So whenever you're doing the plant growth experiment, you're not growing the plants in like a sterile chamber, right? So how do you know part of that effect, that like positive growth effect, is not from organisms in the air depositing on the soil and then responding to those vesicles and then causing the, like, have you done like a environmental microbe control?
We didn't focus on that so much, but we tried our best to sterilize the growth chamber. Every other day, we visited the growth chamber, tried to sterilize it using 70% alcohol. And part of the question you asked is the reason why we're looking at the phyllospheric microorganisms that are associated with the plants above the ground. But regards to the soil, we're almost certain that no extra microorganisms were introduced from the environment, except through the mutualistic interactions in the soil between the extracellular vesicle and the plant roots. No, we're certain about that.
Okay, thank you.
You're welcome.
Okay, I'll turn it over to Marta then. Thanks, Chiquita. And thanks, Matt.
You're welcome.
All right. Hi, everyone. Well, they get to do the hard stuff and I get to do the fun stuff. So mine is my aspect of the project is to incorporate the educational aspect, the K-12 aspect into this research project. And we called it Bacteria Chat. So I'm going to show you how we did that and hopefully give you some ideas of how you can do it in your research project. So the goal, as I mentioned, was to develop an educational component for the broader impacts aspect of an interdisciplinary research project. And if you want to do this, the things that you have to think about are who your audience is going to be. So let's say you're writing the NSF proposal, you get to the broader impact section, and you need to put something in there. So let's focus on education, which is the most important part. So what do? So you think about first who your audience is going to be. And the best thing for this is thinking about what audiences are already existing. So if you have connections, you want to think about the suitable age. And then very important, you want to think about what aspect of your project aligns with any kind of science standards that audience, if you're going for K through 12, is going to be studying. So you could also be doing the general public as an audience, but we're focusing on the K through 12 audience. So then you have to think about among all, and this is sometimes the hardest for a scientist, among all the things that you are doing in your research project and why you think your research project is important, kind of distill it to the most important message that you want to convey to the public or the students about it. So why should anyone care about your research, basically? Who cares about it? Think about where you're going to do it. So to make your life easier, because sometimes it's hard for, scientists have these great ideas and then they want to go into a classroom and they think that the teachers are just going to be really excited about them coming in with their research. And a lot of times they're not going to be that excited because it's so specialized that it doesn't really, they can't see how it relates. But however, the good news is that no matter what you do, we can always find a way to relate it to K-12. So you just need to think about what your platform is going to be, where is there an event that is already taking place that you can fit this into. If you have a school that you work with, or there's an event at the university that brings kids in, these are the things that you want to think about when you're writing your this section. And then how is obviously like how you're going to do it. And one of the things you also need to think about is how you're going to involve the undergrad or grad students in your institution. So in terms of the how, it's really important to get buy-in from teachers or from informal educators, wherever you're going to present your, or wherever you're going to pilot your activities or your educational component. And part of this buy-in is aligning it with the state standards. So I wanted to give a shout out to Allison Gonzalez, who's an undergrad here in our project. She made these really cool bacteria crochet models. And they were a big hit. They're like these big bacteria. I just basically said, hey, make some bacteria out of crochet, and she made it up. So it was really cool. All right, so when you want to, so in order to get buy-in from teachers or from schools for your research project, to implement the educational aspects of your research project, you want to think about how it aligns with their standards. And in North Carolina, we have a set of science standards for every grade, and this is what teachers should teach students at each grade level in science. So for example, if you want to work with second graders and you want to do biotechnology, they don't have biotechnology in second grade, but it fits in perfectly with 8th grade. Or if you want to do rocks and minerals like we do, you might have a friend teaching in a 5th grade classroom, but they don't do any rocks and minerals. They do ecosystems. So just knowing how it fits into the standards. And the reason this is important is because the teachers have a really limited amount of time that they have to teach a huge amount of standards. And unless you show them how what you're going to do in their classroom is going to help align or check off some of these standards, they're not really going to be very likely to invite you into their classroom, unfortunately. If you're doing it with an informal group or a homeschool group or a private school, that's totally different. You could do whatever you want. But if you want to get funding, you're probably better off focusing on a public school. So this will play a role into it. So for example, we wanted students to understand the different ways that organisms sorry, I have a typo there, communicate with each other. We wanted to understand the communities of organisms that make up soil ecosystems, what vesicles are and why study them, and how and why bacteria communicate with each other with plants. So basically, I took what all of these great researchers are doing, and we said, okay, what is the most important thing you want people to get out of your project? And these were the 4 things that we came up, more or less the 4 things that we came up with together. And then I said, okay, well, this fits really well in with the ecosystem unit in 5th grade. So we're going to focus it on 5th grade. It also, even though North Carolina does not adopt the national science standards, they're called the next generation standards. If you're doing an NSF project, because it's nationwide, you want to try to also incorporate how it fits into the national standards so that other institutions, if they want to do something similar, they know where it fits in. These are the 4 students that basically developed the activities with me, piloted all of the activities, and also went into the classrooms and facilitated the activities. And the great thing is that a couple of these students actually did the education aspect for in one semester, a couple of semesters, and then the next year they're actually working with Matt, and they got some experience with the research aspect of it. So their role was to develop the activities and facilitate them, and they work with the Duke grad students to get trained on this topic that they knew absolutely nothing about starting. So we had some grad students in the Duke team that trained them for 2 sessions about all these topics and kind of distill the science for them. All right, so let me give you an example of the activities we did. We had an in-class component and an outdoor component. So we kind of tried the 2 sets of activities. These were pictures from the 4 5th grade classrooms we piloted these activities in. I would say we probably reached about 80 kids, in maybe 80 to 100 kids inside their actual class. We had 2 lessons, so 1 lesson first and then a follow-up lesson. And we did all kinds of things, including group discussions about how different organisms communicate. We had all kinds of models like the crochet models, but also these water beads that really represented the vesicles pretty well. We had microscope work. We created a soil food web card game so they could see where these organisms fit into the bigger food web, because they probably aren't familiar with the tiny microorganisms in the soil, but they're familiar with things like worms and molds and things like that are above the soil. And then we had a vesicle cargo modeling activity. So it's like a game where they could figure out which cargo each vesicle had and how that translated to an actual impact on the organism that was receiving the vesicle and the cargo. And we had pre and post activity assessments to see if they actually learned anything. And this is an example of, this shows one of the students within the vesicle cargo activity and some of the work, what example of a student response from it. And then we did the outdoor activities. So the outdoor activities were a great testing site for these, because we had access to 400 400 5th graders over 2 days. And we didn't have to organize it because it was already organized for us. So that's the kind of thing that you want to look out for. What kind of events can you incorporate your activity into? So we showed up as a 2-day event. It's organized by the Soil and Water District Office in Watauga, and we were in Valley Cruz's community park. We set up a table there, and the researchers from App State and the students were the ones facilitating these activities, which was really neat. So we had really good feedback, and we had a soil ecosystem diorama. You can see Rachel down there discussing it with the kids. We also had vesicle micrograph kaleidoscopes that we made out of Pringles cans. So, you know, we couldn't have the high-powered microscopes to see this, but they really enjoyed looking into the sun and looking at these kaleidoscopes that we made with a real micrograph that I think Hannah took. So this was an actual micrograph with fake colors for the kaleidoscopes. And then on the top there, we have a jumbo role play. So we basically set up ropes to show 2 bacterial cells and the membranes of the cells. And then there were these plastic spheres all over, and there were the vesicles. And we had the kids communicate with each other and pass the vesicles back and forth. And then we had a system of scoring where, like, if your bacteria cell received vesicles that had a cargo that were beneficial for the cell, they had positive points. And then if they had like an antibiotic that would harm the cell, they had negative points. And then they did a bunch of math, so it incorporated math as well to figure out what team won. Other than the pilot activities, we also we created a website to kind of serve as a hub for everything that we produced. And the website not only includes just the general language or general layman's language for like what vesicles are, but also all the activities that we develop. So there's links to all the activities that the teachers can download and try in their classrooms. And all our materials were in English and Spanish because we had 3 Spanish speakers in the team, so we translated everything. And here are some examples of some of the activities on the resources. So some amazing artwork by Hannah, who is one of the grad students in the Duke team, so multi-talented scientists, and some examples of some of the handouts that we had, that we created. And what were the learning outcomes? So we loosely did some post-assessment, especially in the classroom setting, because the outdoor setting was a little hard to get some really good assessment down. But basically, we had students that went in thinking that vesicles were little blood veins or little worm-looking things, tentacles, a cooler for a greenhouse to keep the plants from drying up, because it has part of icicle in the word, and vessels that soak up the sun. 2, saying things like vesicles are packages with chemicals in them, small fluid-filled sacs, packages of chemicals used in positive, negative ways produced by cells or bacteria. So these are 5th graders who are about 10 and 11-year-olds. And here's an example of the pre and post assessment like collection that we did. And some pictures as part of the post assessment as well of all the vesicles and the bacteria, passing them back and forth. And another one. These were pretty awesome drawings. And that is it. I went super quick because I'm like worried about that I was going to go over, but I think it's great because I have to leave at 2. So if anyone has any questions, we have a little time for questions. It was great to collaborate with all these scientists. I really enjoyed the multidisciplinary aspect of it, or also the cross-institutional aspect of it. I want to thank Maida for supporting me and endorsing me to go to this American Society for Intercellular Communication conference. My poster was definitely very different from all the others. No one had plastic vesicles and crochet bacteria. These were all like on both sides. I had like AIDS vaccine research and like all these different like childhood disease and things, like, I mean, amazing science. And then I had my little toys, but they were great. And just like, I hope this inspired you to maybe get some, give you some ideas for your next research project for that section that, sometimes people have a hard time coming up with things that are realistic for. So thank you. And thanks to Rachel and Matt for your hard work. And yeah, any questions, let me know.
Thank you, Marta. You landed just on time. If you want to stop screen share so we can see one another, we can open it up for discussion, question and answer. And as Marta said, she has to leave at the hour, but Rachel and Matt can stay on. So if there's good conversation going, feel free to stay beyond that. And I'll go ahead and open it up for questions. Thank you all.
I'll ask a quick question. The activities look super fun. I always love the hands-on activities with that age. And I'm curious how, like, if your plan is to kind of make these lessons public so teachers can use them, or do you see it as more of a like partnership? We come in, kind of teach, or you go in, kind of teach, and I'm just curious what you see kind of the long term being.
Yeah, that is actually a great question, Kimberly, because a lot of times we produce these resources for grants and they really don't get any kind of views or anything, and you put so much work into it, but really, and I know going in that is going to be like that, but I saw it as a way, like if anyone, like when I presented at this conference with all these vesicle scientists, that they will get some ideas for when they have to either write another grant or go into a school to do a presentation. If I wanted the teachers to use it, which I do obviously, but you really have to put in a lot of legwork to get your work out there. So for this one, I can't remember if we had as part of it to present at a teachers conference, but that would definitely be a component that you would want to put in if you want it to be widely disseminated. So for other grants I'm on, this is a much bigger piece. But yeah, that's a really good question. If you want your work out there, you really have to put in the light work to advert, because there's so many resources out there. Yeah, so, but good question. And if anyone, and this is something that this is obviously something that not a lot of people that are scientists are going to have the expertise to be able to align standards to their research. It takes understanding these. So you have to seek out people like me who are also straddling the education world, who can say, hey, you're doing this for whatever topic. It doesn't have to be science. It could be anything. And then they can align it with the standards or with a grade level that is appropriate.
I have a question while we still have you, Marta, is I got an opportunity to come to part of the 5th grade bonanza, and I have a 5th grader, so he gave it on high authority that it was the favorite event of the day, and he was ready to be embarrassed by me, so I know he's telling the truth. So it was such a brilliant way that you made a complex thing simple. And one of the reasons that we wanted a younger age range of outreach, originally when we were writing the grant, before we knew anything real, which you showed us, was because we thought that if people are talking at a level that they could communicate with somebody in elementary school, then they might not have a built-in assumptions that would keep them from, as scientist to scientist, communicating across disciplines. So when you went to the science conference with Marta, what were some reactions that you had from these science folks about it? Like, did you, I'm curious how it went over with them and how it was different, like what ahas did you see in them?
Yeah, well, first, and I think Marta might be able to agree with this, is like most of them just had no idea or didn't even really think about vesicles in the environment. So it's like they're so specialized that even something that is so, like they didn't, so even that was, I think on the but yeah, they didn't do anything like this. But I think maybe a lot of the grants are mainly through the NHS, right? Like they're not like NSF ones, so they might not have like this as much of a focus maybe needed on broader impacts or education, because you obviously have the broader impacts of the health stuff. But I don't know. Yeah, it was definitely they seem to enjoy it, but who knows, maybe it was because it was such a change from like, what they were doing. But we do, I mean, we get funding with this. So we've gotten a ton of grants with, so I think we have some, we have it down. So if anyone needs any help with this or ideas, I'm happy to talk. I mean, we don't always get them funded, but yeah, seems like it's great for people to actually get out of their region and like have to answer the question, like, why would anyone care about what you're doing? So I asked them, it's like, our tax dollars are funding this. Like, why would we care about it? And it just makes you think about what's really important about your research.
It looks, Meta, you have a question?
Oh, I was just going to say, I think Marta's poster is a big hit, especially, which was very, I think, heartening at the conference with the younger scientists there, like the graduate students and the postdocs who, I mean, this totally opened their eyes to a different aspect of what they're working on every day, day in, day out in the lab with these vesicle purifications and all these little tiny assays and, test tubes of, invisible molecules you can't see, and that they're, transferring, looking at effects on host cells and that kind of thing. And to see somebody be able to communicate what they're doing at this nanoscale level in a macro way to children and have them get it, and at such a fundamental level. I think it was eye-opening for people at that scientific meeting in a completely different way, sort of, a groundbreaking way. So I think it was a great success.
What a neat thing to witness, a nice outcome, I guess, to see their work made real in an eye-opening way.
And it was just as beneficial for me because, someone that is more in the education aspect, to be able to experience all the research that's going on and kind of how we could, inspire kids is also really useful. So it gave me a lot of ideas too.
Yeah, Steve, you had a question?
Yeah, I had a quick question, primarily for Matt and Rachel. So, first of all, congratulations. This is actually kind of interesting work here, and the implications are obviously very broad. Am I correct that when you isolated vesicles, you just isolated all vesicles?
Correct, yeah.
I know nothing about these little vesicles, so I may be barking up the wrong tree. But at the same time, if you're isolating all vesicles, do you not have a mix then of vesicles that could be have positive and negative effects to the plant and to other bacteria? So, I mean, you got a response, obviously, in terms of plant growth, but how do you attribute that to specific actions if you're looking at a mix of vesicles carrying different chemical characteristics and signals?
Yeah, that's a great question. Thanks, Steve. You're right. When we are isolating these vesicles, it's just like a heterogeneous population of everything that's budding off. So very much dependent on the conditions the microbes were grown in and kind of what stressors they're under. So right. I think further follow-up studies of exactly like what cargo is in there. And I know there are some people working on ways where you can further like try to fractionate a little bit more by size. So you maybe can try to get a more consistent pool of macromolecules that are in the cargo. But you're right. It's just kind of like a broad swath.
Yeah, you start somewhere and then start working forward from that. And, the ultimate thing is then trying to fit this type of work into the complexity that you find in a real soil ecosystem, and the thousands of types of bacteria and things like that are going to all create different vesicles. It's a complex world out there. It's nice to understand a little piece of it.
Right.
Steve, how did you landed us on a perfect summary sentence. It's a complex world out there. It's nice to understand a little piece of it. We don't have to disband now, but I wanted to say thanks to Marta, and I know she's got to take off at the hour, and for anybody else who can't stay for a discussion Q&A, just thanks for joining us today. And the next one we have will be March 3rd, and it will be the history of the CDAC carbon emissions database. That is the oldest emissions database on the earth, and App State and some of our great scientists here are the stewards of it. So join us for that next time, and I think we'll end it there.